Hello, and today we'll be going through glycogen metabolism, but a little bit more in depth. First, let's review the fates of G6P. So it can either be turned into glucose, or it can go into glycogenolysis, or it can be made into ribose 5-phosphate into the pentose phosphate shunt. So glycogenesis is the creation of glycogen. Usually it is made in the liver and the muscles. Those are the main organs that produce a significant amount of glycogen. And it is made within the cytosol. So there's a couple of unique enzymes that aren't shared with glycolysis, and one of them is the alpha 1,4 branching enzyme. The alpha 1,4 alpha glucan branching enzyme 1 is the full name, and then glycogen synthase. So first, it needs to be activated. So glucose 6-phosphate is transformed by phosphoglucomutase into glucose 1-phosphate, and from there it is turned into UDP glucose by UDP glucose pyrophosphorylase. And then from there, it is turned into glycogen once it's reacted with glycogen synthase and a UDP has been ejected. So the glycogen synthase makes the alpha 1,4 linkages. So these would be the linear linkages, and by the creation of like the linkages and the units, glycogenin is a fragment of glycogen, and that is the primer. So it begins all with the glycogen after activation. Here it goes. UDP has been ejected, and then now it goes into glycogen, right? And then glycogen synthase, UDP has been ejected, and then from the glycogen, like things are attached to it. So UDP is released by UDP nucleoside diphosphate kinase. Remember, kinase is going to be dephosphorylated. Okay, so the more important parts, branching. So Glucosyl Alpha 4,6 transferase, it's in the name. It will be transferring the units from an alpha 1,4 to an alpha 1,6 bond. So it transfers a chain of like about 5 to 8 glucosyl residues from a non-reducing end to another non-reducing end. So it takes the units here, and then it is attached to a branched alpha 1,6 molecule after it has been branched, right? So it is just like it is just used to make the branches, to compact the molecule instead of making it longer and longer, right? So we're going to go through glycogenolysis a little bit more in this part. So the degradation is in the liver and muscle, where it's produced, of course, also in the cytosol, and the main product is a G1P and a free glucose. Okay, so after we have made all of these bonds, as like, the transfer of like, a unit from the main linkage to the alpha 1,6 linkage, we're going to do that backwards in the glycogen degradation cycle. So glycogen phosphorylase only breaks the alpha 1,4 bonds, so it only breaks things in a linear way, and by doing that, it makes a G1P. And the D-branching enzyme, illegal 1,4,1,4 glucan transferase, will break only the alpha 1,6 linkage because it will debranch things, right? So it removes the outer 3 glucosyl residues and transfers it to another non-reducing end to elongate. So it would be chopped in things like right here, and then this would be removed, and then it would be attached here. So now it has been debranched. There, bam. And then amylo alpha 1,6 glucodase will only break the 1,6 linkage, like the actual linkage. Before, the debranching enzyme will only affect like the main branched unit. So it won't actually break the alpha 1,6 linkage. Sorry, I should have been more clear. So it will like cleave it only in the alpha 1,4, but up to the actual branched unit. So finally, it's been transferred. There's one sad little unit left. The amino alpha 1,6 glucosidase will cleave that, and then that will be the free glucose. And then this will repeat, because this is like a very very basic example. Usually there's like branches upon branches and upon branches, so it will go like top, and then elongate, and then like chop it again, and then elongate until it breaks down to like the core. So glycogen synthase is regulated because there's two different forms. Synthase D, this is phosphorylated, and it's usually inactive, and synthase I, or synthase insulin, is non-phosphorylated, and that is active. So these are activated by G6P, and it is degraded by the lysosomal acid maltase. So glycogen phosphorylase, there's two different forms. A, it's phosphorylated and active, and is allosterically regulated in the liver. And there's the two forms of phosphorylase A. R is relaxed, and T is taut, kind of similar to the way hemoglobin is made. So when it's relaxed, things will bind until it's taut, and then it would be activated. It's inhibited by G6P. And then phosphorylase B is non-phosphorylated, and it's allosterically regulated in the muscle, and it's activated by AMP and inactivated by GCP and ATP. So in the muscles, calcium will bind to the calmodulin part of the subunit of phosphorylase kinase, and it will activate the kinase without the CAMP-dependent protein kinases. So it skips over a regulatory step in order for the muscles to kind of like use the most amount of energy, like the most efficiently. So the hormones, glucagon and epinephrine, will activate adenylate cyclase, and it will inactivate glycogen synthase. So makes sense, you know, epinephrine, you want more glycogen to be broken down, not made. And the cascade of reactions that result in glycogen degradation amplify the hormone signal. Makes sense because hormones are kind of expensive to create, so we want the best bang for our buck, so to speak. So only a few hormones will be released, and then once they bind, there would be a cascade, and that will amplify the signal. And insulin, of course, will increase the glycogen synthesis. Okay, so let's go through the diseases. So Glycogenosis type 1 is autosomal recessive. It's also known as Van Dierk's, and it is a pleiotropic, which means one gene will affect several different like phenotypic manifestations. It's caused by these point mutations and splice site defects. And these are the clinical features: short stature, liver and kidney enlargement, fasting, hypoglycemia, hyperlipidemia, and hyperuricemia. Other diseases that are associated is the liver adenoma and osteoporosis, but this is the main clinical feature, the chronic renal disease. Another form of it, the GSD1B, would form more of like a immunodeficiency. So there would be neutropenia and neutrophil dysfunction. So the immune system would be less effective, causing the recurrent infections, and then they would have IBD. And look here, this poor kid has all this ascites, and look at these like thin purple lines. These are called striae, and striae is pretty characteristic for liver enlargement or hepatomegaly. Okay, so type 1A is a GTPase defect, and type 1B is a glucose 6-phosphate translocase defect. And these are the clinical features again: growth retardation, muscle weakness, liver cancer. Unfortunately, the patients that suffer from this disease, they don't live very long. Glycogenosis type 3 is also known as Cori or Forbes disease. And there is going to be defects in the branching enzymes, amylo-1,6 glycosidase. So this would interfere with the alpha-1,6 glycosidic linkage. So the gene is found on chromosome 1p21. There are 35 exons that code for different tissue types, and it is is characterized by hepatomegaly and hypoglycemia, and it is usually found in North African Jews because of the founder effect. And it is caused by a frame shift mutation on the C-terminal, and it causes a truncation of the protein. So what's important to know about all these glycogen-related diseases is, well, why does it cause hepatomegaly and hypoglycemia, right? Like, what does the branching enzyme have to do with anything, right? So hepatomegaly is because of the abnormal glycogens. They take up too much room. Remember, branching is to compact the molecule. So without it, it is too large and it's abnormal, and it builds up in the liver, and the liver has no choice but to get bigger and bigger to try to accommodate for all of that. Why hypoglycemia? Because none of the glycogen is being broken down. All it's doing is being stored away. So look at this poor child here. Look at these lines, right? This shows the enlargement of the liver, right? Is ridiculously large. The liver should be maybe a third of the size, but it goes all across the abdomen. All right, these are the other diseases. So type 4 is also a branching enzyme defect, and it's found on chromosome 3p12. It's also known as Anderson's disease. It's an early onset, also has the hepatic and spleen cirrhosis features, also includes cardiac and neuromuscular diseases, and unfortunately this is fatal by age 2. Type 5 involves muscle phosphorylase on chromosome 11p13. This one's not too bad. This just means that patients that have this deficiency just have a low exercise tolerance. They get fatigued easily, they get winded easily, and they'll be fine after taking a little break. So type 6 is involved in liver phosphorylase, which is found on chromosomes 14A21 to Q22. It's also known as Hurler disease, and this is a strange disease because the first few years, up to a decade, it's characterized by liver disease, so hepatomegaly, right? And then over time, it just resolves. It's not really known why, but it just resolves with age, and the liver goes down, the hypoglycemia resolves, and then there's no more ketosis. But in the meantime, while the patient is suffering with the hepatomegaly, it is important to treat these diseases. So type 7 is involved with muscle phosphofructokinase, and that's found on chromosome 1sen all the way to Q32. It's also known as Tourette disease. It has similar features to type 5. It's just caused by a different chromosome defect. So type 8 doesn't have a name. It's just involved with phosphorylase kinase, and it's on the chromosomes XQ12 to Q13, and this is a slow progressive muscle weakness and then atrophy. Okay, here's a table that kind of involves all of this. And these are all the tissues that are involved. So just remember what part of the glycogen metabolism is being effective, and what enzyme is being affected, because there's a couple enzymes that only exist in like liver or muscle, right? So it's kind of important to know those. Okay, so let's go through galactose. Here's galactose versus glucose. As you can see here, galactose, the hydroxyl group is above the plane. Okay, so most of the source of galactose is actually from dairy. It's beta galactosidase, also known as lactase, is the enzyme that metabolizes it. It is not insulin dependent, and it's a slow degradation because it depends on phagocytosis. So once you've eaten the lactose or galactose, the body has to absorb the molecules, and then by doing that, it has to phagocytose, so it can't just freely diffuse in. So D-galactose is phosphorylated before anything starts. This is our activation or primer step. And then from there, your UDP galactose will make G1P, as you can see here. And it's an expensive process. It takes energy to make G1P. And then from there, G1P is transferred by a mutase into G6P. And from there, it goes into glycolysis. So galactose can be recovered using an enzyme called epimerase. Epimerase. And yep, there you go. And then we'll make the UDP glucose, and then it can be recreated into glycogen. Okay, so galactosemia type 1 involves UDP G galactose 1 phosphate uridyl transferase, and that is on chromosome 9p13. The clinical features would be vomiting, diarrhea, jaundice, liver disease, ascites, cataracts, mental retardation. Type 1 is the most dangerous of all the diseases because the galactose will build up in the blood. And then there would be liver failure because the liver is going to try to compensate for that. And then by doing that, it will push out all this water, causing fluid buildup in the abdomen. And type 2 is galactose kinase involvement, and that's on chromosome 17, Q24. And in this disease, their cataracts are formed because one of the metabolites, galactitol, that is formed can't be exchanged. It will settle in the eye, and then because there's more molecules in the fluid, osmotic pressure will increase, and then in order to kind of save the eye, the body will replace the kind of the I want to say soft collagen in the eye. It will be replaced with fibrous tissue, so that will cloud the eye. So type 3 is UDP galactose 4 epimerase, and that's found on chromosomes 1p36 to p35. There's two different forms. The benign form, where the enzyme deficiency is limited to only red blood cells and white blood cells. This is an asymptomatic form. And the severe form has the transferase deficiency with hypotonia and sensor neurodeafness. And so, of course, it can't be galactose. UTP can't be removed, and then the galactose can't be turned into G1P. So the muscles will be starved of energy, causing the hypotonia. And then the neurons in the ears require a lot of energy too, so there would be degeneration because of the lack of energy. Right, so lactose is made by lactose synthase, and by doing that, it releases a UDP. And then there's two different types. It can go into protein A is made in all the tissues, and protein B is only made in the mammary glands, and that's controlled by progesterone. So progesterone in females will inhibit protein B synthesis, and until she is, breastfeeding, prolactin will come and then activate it at that time. So these are the paths that lactose can take. It can go into, it can be made into galactose, and then from there, it can go into here. Oops, I'm sorry, keep doing this. Or it can go straight into glucose, and then that will go into the PPP, or it goes into glycolysis, or it can go into glycogen, and then over here, it can even go into like the fructose pathway and be made into fatty acids, or it can be made into it can be made into the N-acetylglucosamine 1-phosphate. Okay, so lysosomal storage diseases are genetic defects in the lysosomal enzymes, and those defects will cause an accumulation of the substrate, which will lead to cellular dysfunction and cell death, and that is generally characterized by neurodegeneration. Okay, so Pomps disease is a type 2. There are defects in acid maltase, and that enzyme is it's encoded on chromosome 17q25.2 to q25.3. The infantile type A appears within the first 2 to 5 months, and then unfortunately it is fatal by age 2. It's characterized by severe muscle weakness, enlargement of the heart, liver, and tongue. Childhood type B, muscle weakness, especially in the respiratory muscles, and it's fatal by their second decade. And the adult type C is similar to type B, so the respiratory muscles are involved, and the death will be in the 4th decade. Okay, so Mucopolysaccharidosis is a group of lysosomal storage diseases, genetic defects of which involved in the glycosaminoglycan degradation. So this is found in the extracellular matrix, and the normal role of this enzyme is to break the protein down into its constitutive amino acids. And this will affect the bone, the viscera, the connective tissue, and the brain, because this is where a lot of energy is stored, right? So the mucopolysaccharidosis involves the urinary excretion of abnormal mucopolysaccharides, and mucolipidosis is the urinary excretion of normal mucopolysaccharides. So this is a little bit more dangerous because you're excreting all your normal polysaccharides. This is dangerous because this shows that the body is making some abnormal mucopolysaccharides. Okay, so Hurler Syndrome is involved in MPS IH, and that is the prototypic mucopolysaccharidosis. It is an autosomal recessive inheritance pattern, and there's going to be a defect in alpha L iduronidase, which prevent the normal catabolism of dermatan sulfate and heparin sulfate. It's characterized by the dysostosis multiplex, the hepatosplenomegaly, that's important, claw hands, the coarse thickened features, there you go, the corneal clouding, you can see valvular problems, pulmonary hypertension, also known as gargoylism. So it's a combination of the clawed hands, the coarse features, and the scoliosis. So what's important to know is the dermatan to heparin ratio, sulfate ratio, is 7:3. So they don't tend to live very long either. Usually they die of either the hepatospleno disease course, or pulmonic and cardiac involvement. So Shea and Hunter diseases are also defects in iduronidase. In the Shy or the Shay disease, there's going to be a defect in the alpha L iduronidase, and allelic mutation is all at the same locus. This one also shares the coarse features with the Hurler's disease, the corneal clouding, the cardiovascular disease, stiff joints, and genu valgum. What's important to know is that the dermatan to heparin sulfate ratio this time is 3:2. So before it was 7:3, now it's 3:2. Hunter's is an X-linked deficiency in iduronate sulfatase. And the phenotype is also similar to Hunter's, but this one has no corneal clouding. Instead, there is deafness. It also has some mental deterioration, respiratory disease, and cardiovascular disease. The dermatan to heparin sulfate ratio is going to be 1:1. So, complementation analysis is used to find the genetic loci to see whether these diseases are going to be manifest in your child. So the cells are able to export tagged enzyme. The correction of both biochemical defects of both cells are noticeable. So both cells will have the defect, right? And then if you're able to find that loci, you can potentially use gene therapy or enzyme therapy to treat the disease. Okay, so transport disorders are generally like lysosomal disorders. The eye cell disease is the lysosomal enzymes that exist in the cell but are not able to reach the lysosome. So the lysosomes are essentially useless. They fail to target the acetylglucosamine 1 phosphotransferase. And by that, there's not going to be any binding, right? So without the binding, it's not going to be able to enter the cell. This is an important transferase because it adds the mannose-6-phosphate onto the protein target. And from there, it's able to recognize that, hey, we need to kill this. And there would be a problem in that kind of recognition in this disease. It forms in the cytoplasm, and in these patients, there would be the severe mental retardation, the skeletal abnormalities, respiratory infections, cardiorespiratory complications, and unfortunately, there would be a death within 10 years. It's more because of the recurrent opportunistic infections. So, as you can see here, it's all sorts of disorganized, right? And then there's all of these like floating materials that should be in the lysosomes. All right, and that's it. Thank you for paying attention. Good luck studying.